Hello everyone, welcome to MBBS Atlas. In this video, we are going to talk about fibrinolysis. Now, in the last video, we have spoken about blood coagulation, right? What exactly happens in blood coagulation? The fibrinogen. So the fibrinogen, it is converted to fibrin, right? And this fibrin, it forms the tight blood clot. Okay, now what is the exact meaning of fibrinolysis? See, as the name suggests, fibrinolysis, that is lysis of fibrin. Okay, so breakdown of this fibrin, that is the dissolution of blood clot, is simply known as fibrinolysis. So it can be defined as the physiological process by which fibrin clot formed due to coagulation is broken down once healing has occurred. Okay, now this fibrinolysis, it ensures that blood vessels remain open and prevents excessive clot formation, that is thrombosis. So what is the main agenda or the main significance of fibrinolysis is to prevent the thrombosis. Okay, and fibrinolysis requires a substance called plasmin or the fibrinolysin. Okay, now we'll study the formation of this plasmin. Now plasmin is an activated form of plasminogen. Okay, so plasminogen is an inactivated form of plasmin. Okay, and this plasminogen, it is a glycoprotein and it is secreted from liver. So it's a glycoprotein. Okay, it's a glycoprotein which is inactivated form of plasmin. It is inactivated and it is synthesized from the liver. Okay, so this plasminogen is activated with the help of some activators for the formation of plasmin, and this plasmin causes the lysis of clot. Okay, now we'll be looking at the activators of plasminogen for the conversion of plasminogen to plasmin. Now what are the activators? The first one is TPA, that is tissue plasminogen activator. This tissue plasminogen activator, it is released from the endothelial cells, and remember always, this TPA is always present in inactivated state in our blood. Now the question rises, why this TPA is present in inactivated state in our body? Because of presence of something called as TPA inhibitor. Okay, because of this TPA inhibitor, factor 5 and 8. So these keep the TPA in inactivated state every time. So to activate the TPA, first we need to inactivate the TPA inhibitor and factor 5 and 8. Okay, are you getting my point? See, this TPA is present in inactive form always. Why it is present in inactive form? Because of presence of this TPA inhibitor, factor 5 and 8. So for the activation of this TPA, we require the inactivation of these things. So for the inactivation of TPA inhibitor, factor 5 and 8, we have something called as protein C. We'll be studying this again. So we'll be having something called as protein C. This protein C, with the help of protein S. So protein C with the help of protein S, this protein S is a cofactor for protein C. So this protein C with the help of cofactor protein S, it inactivates the TPA inhibitor, factor 5 and 8, causes the activation of TPA. Okay, I hope you are getting my point. If you have any doubts, please do comment. Okay, the second one is the UPA, that is urokinase plasminogen activator. So it is formed in plasma and tissues. Okay, now the factor 12 and calcarein, these are also one of the activators of plasminogen. And the last, the lysosomal enzymes and thrombin. These lysosomal enzymes and thrombin are released by the damaged endothelium. Okay, so these are the four main activators of the plasminogen for the conversion to plasmin. Okay, what are the activators? Lysosomal enzymes, thrombin, which are released from the damaged endothelium, TPA, UPA and factor 12, activated factor 12. Okay, now we'll dive into the detailed process of this reaction. So this reaction has a very detailed process in between, so we'll be studying it. Okay, see the sequence of events involved in activation of plasminogen. See, when there is tissue damaged, there will damage to endothelium, then lysosomal enzymes are released. Along with the lysosomal enzymes, there is also release of thrombomodulin and thrombin. Okay, thrombomodulin and thrombin are also released along with lysosomal enzymes when there is damage to the endothelium. Now this thrombomodulin and thrombin, they combine to form thrombomodulin thrombin complex. Okay, so they combine to form thrombomodulin and thrombin complex. This thrombomodulin and thrombin complex activates the protein C. So this protein C is first present in inactivated form. So from inactivated form of protein C to the activated form of protein C is converted by the thrombomodulin thrombin complex. Okay, now protein C is activated. Now after the activation of protein C, with the help of protein S, it inactivates factor 5, 8, and it also inactivates TPA inhibitor. As we have studied here, it inactivates factor 5, factor 8 along with TPA inhibitor. Now the TPA inhibitor, factor 5 and 8 are inactivated. So now the TPA gets activated. So the tissue plasminogen activator gets activated. Now when this is activated with the help of thrombin, UPA and the lysosomal enzymes, with the help of lysosomal enzymes, UPA and thrombin, the plasminogen is converted to plasmin, and this plasmin causes the lysis of clot. I hope you are clear. First, thrombin thrombomodulin complex is formed. This activates the protein C. Protein C, with the help of protein S, it causes inactivation of TPA inhibitor factor 5, 8, which in turn leads to the activation of TPA. With the help of thrombin, UPA and lysosomal enzymes, TPA causes the conversion of plasminogen to plasmin, and plasmin finally causes the lysis of the clot. Okay, I hope you are clear with the mechanism of fibrinolysis. Now we'll be studying a reasoning question, which is a very important reasoning question. So the question goes like this: why does blood does not clot in vivo? Now think of this: why does the blood does not clot in vivo? So now let us look at the factors preventing blood clotting during circulation. Now the first one, the smooth surface of endothelium of blood vessel prevents the activation of clotting factors. Because of the smooth surface of endothelium of blood vessels, clotting factors and the platelets are remained in inactivated state. Okay, and the second one, continuous flow of blood does not allow aggregation of platelets and prevents blood clotting. I hope you got this point, you understood this point. It's a very basic point. And the third one, the glycocalyx layer on the inner surface of endothelium repels platelets and clotting factor, preventing the blood coagulation. See, we all know that we have endothelium. The inner surface of endothelium, it has a glycocalyx layer, and the nature of this layer is the repelling nature. It repels platelets and clotting factors. Obviously, when platelets or clotting factors are not together, they do not form a blood clot, right? Next, fourth one is the presence of natural anticoagulants in blood. What are the natural anticoagulants? Heparin and protein C. Now, heparin is an anticoagulant produced by liver. Okay. Next, protein C. It is activated by thrombomodulin thrombin complex. As we have studied here, it is protein C is activated by thrombomodulin thrombin complex, right? So it is activated by thrombomodulin thrombin complex. So now the activated protein C along with cofactor protein S, it inactivates factor 5, 8, preventing the blood coagulation. I hope you got my point, and here ends our video. If you have any doubts, please do comment in the comment section, and thank you for watching this video. Bye.